Without further ado, hello everyone. Welcome to the ATS Peds Assembly Journal Club on the Transcriptomic Profiles and Asthma Endotypes in Pediatrics. I'm Dr. Annie Coates from the Division of Pediatric Pulmonology at the Barbara Bush Children's Hospital in Portland, Maine, and I'm delighted to be co-moderating today with my colleague Dr. Tang Santiago from the Division of Pediatric Pulmonology at Cohen Children's Medical Center of New York. Today we'll be addressing transcriptomic profiles in nasal epithelium and asthma endotypes in youth, published in JAMA in January of this year. Dr. Kristina Gaietto, my co-chair of the Peds Assembly Web Committee, is an instructor of pediatrics at the University of Pittsburgh School of Medicine in the Division of Pulmonary Medicine. Her research has focused on the impact of psychosocial stressors on asthma and asthma outcomes, SARS-CoV-2 infection on children with asthma, and asthma endotypes. She is one of the authors of this article, and she will be presenting on it today. In addition to Dr. Gaietto, we are honored to be joined today by our panel of experts who are well known in the field of pediatric pulmonology. Dr. Len Barcharier is the Janie Robinson and John Moore Lee Chair in Pediatrics and Director of the Center for Pediatric Asthma Research at Vanderbilt University, where he is a professor of pediatrics, allergy, immunology, and pulmonary medicine. Dr. Barcharier has devoted his career to research to help understand and improve the care of children with asthma. His clinical and translational research efforts are directed at the pathogenesis of asthma in early life and approaches to asthma management throughout childhood, including multi-center, federally funded clinical trials in asthma. Dr. Juan Celedón, also one of the authors of this Journal Club article, is the Niels K. Jern Professor of Pediatrics and Medicine at the University of Pittsburgh and Division Chief of Pulmonary Medicine at UPMC. His research goals are to identify genetic and environmental determinants of airway diseases, particularly in racial and ethnic minorities. Dr. Celedón leads NIH-funded projects on the omics and epidemiology of asthma. Dr. Deepa Rastogi is professor of pediatrics, pathology, and genetics at the Albert Einstein College of Medicine. She is the division chief of the Pediatric Respiratory and Sleep Medicine Division at Montefiore, Einstein. Her federally funded research investigates the mechanisms that underlie obesity-related asthma. Her clinical specialty is in taking care of children with asthma, particularly those with severe or difficult to control asthma. Dr. Kelan Tantisira is chief of the Division of Respiratory Medicine at Rady's Children's Hospital in San Diego and a professor of pediatrics at the UC San Diego School of Medicine. His primary research focus is on the factors influencing asthma susceptibility, severity, prognosis, and treatment response, with particular interest in asthma pharmacogenomics, translational genomics, and personalized prognostic medicine. Talk about an all-star lineup that we have here. So without further ado, Dr. Gaietto, please take it away.
Thanks, Annie, for that kind introduction. And thank you for the opportunity to present today. Thank you to the panelists for joining and for all of you for logging on to listen. I'm excited to get to talk to you about our study. All right. So as far as conflicts of interest go, just for everyone to be aware, Merck provided Asthma-Nex given to participants in one of the studies that we included in this study. And then Pharmavite provided vitamin D and placebo capsules for participants in VDKA, which is one of the studies we included in this study. And then GSK provided the Flovent that we used in VDKA. And then our funding sources are listed here. And just to be clear, this is not a study of any medications. We'll get to what the study's about, but I just wanted to be transparent about those disclosures. So for today, the plan is to go over some background to give some context, then to go over the article itself, and then we'll go into the discussion with the panelists and have an opportunity for Q&A. So we'll start with a little bit of background. Asthma is a heterogeneous syndrome driven by different pathogenic mechanisms or endotypes. Initially, two different endotypes were described based on the presence or lack of T helper 2 cells, which are a type of CD4 positive T cell. Individuals with T2 inflammation were categorized as T2 high, and individuals without T2 inflammation were categorized as T2 low. And it was initially thought that most asthma was T2 high, while a minority was T2 low. Subsequent studies started to elucidate details of the inflammatory pathways associated with T2 high asthma. So T2 high asthma is associated with eosinophilic airway inflammation and/or allergic airway inflammation. In contrast, T2 low asthma is not as well understood as T2 high. We know now from prior studies that T2 low asthma can be divided into T17 high and T2 low, T17 low asthma. T17 high asthma is characterized by neutrophilic airway inflammation and increased levels of IL-17 and IL-22. The T2 low, T17 low endotype, which is paucigranulocytic, hasn't really been very well characterized yet. And some of you might be wondering why we care about this or why endotypes matter. An individual's asthma endotype matters because it influences treatment approach, particularly for severe asthma. We have some monoclonal antibodies that can be used to treat severe asthma, but all but one of them is only for T2 high asthma. These T2 high asthma biologics include dupilumab, omalizumab, mepolizumab, and benralizumab, while tezepelumab is the only biologic approved for any type of severe asthma, regardless of asthma endotype. And tezepelumab currently is only approved for use in ages 12 and up. The other biologics that I mentioned can be used for severe asthma in children as young as 6 years old. But there are currently no biologics approved for treatment of severe asthma that specifically target these T2 low asthma pathways. So the huge problem is that it can be really hard to determine what asthma endotype an individual has, especially in the clinical setting. There's some biomarkers that we use for T2 high asthma, which include total IgE, FeNO, blood eosinophils, and sputum eosinophils, but there's not a consensus for what cutoffs to use for these. Sometimes allergic sensitization is used to define T2 high asthma, but recent evidence suggests that atopy isn't synonymous with T2 high asthma. People with T2 low endotypes can also have atopy, and there are no serum biomarkers or lung function tests for either of the T2 low asthma endotypes. So you can see there's a desperate need for better ways to identify asthma endotypes, for research studies, to develop targeted treatments, and for clinical practice. As a pediatric pulmonologist who cares for kids in the severe asthma clinic here in Pittsburgh, I can tell you that this difficulty identifying asthma endotype is extremely frustrating. Often I have patients who have borderline markers of T2 inflammation. Some of these patients respond to the T2 biologics while others don't. And if a kid gets oral steroids for an asthma exacerbation, which happens really often in the severe asthma population, then their labs can be unreliable for a few weeks. So we have to wait to try to assess their endotype. All the clinicians here know that after a kid gets systemic steroids, their CBC is just going to show a ton of neutrophils and likely not many eosinophils. And FeNO can be impacted by dietary nitrates. So if a kid eats a hot dog for lunch before their FeNO test, their results could be altered. So our existing methods to assess T2 high asthma in clinical practice aren't optimal. And we have nothing in clinical practice to identify T17 high or T2 low, T17 low asthma. All we can say is that a child doesn't seem to have T2 high asthma, so by default, they probably have some type of T2 low asthma. So that brings me to our study. This is a study of using nasal epithelial gene expression to determine asthma endotype in youth. I want to highlight Mo-Lin Yu, who is a brilliant bioinformatics PhD candidate, who was co-first author on this publication. Mo-Lin and I worked really closely with Dr. Juan Celedón and Dr. Wei Chen, who were the senior authors on this study, as well as our co-authors who are listed here. So how did we come up with this idea to use nasal epithelial gene expression to determine asthma endotype in youth? Over a decade ago, in a seminal paper, bronchial epithelial gene expression was shown to distinguish T2 high from T2 low asthma in adults. And then a few years later, analysis of bronchial epithelial samples from adults with asthma revealed 3 distinct patient clusters: T2 high, T17 high, and T2 low, T17 low. And this was based on expression of 3 IL-13 inducible genes, which are the T2 genes I have listed here, as well as 5 IL-17 inducible genes, which are the T17 genes I have listed here. And really importantly, the group of individuals with high T2 gene expression and the group with high T17 gene expression were mutually exclusive. However, this was done with bronchial epithelial cells, which you have to get invasively via bronchoscopy, and these studies were done in adults. So our group, led by Dr. Celedón, wanted to see if we could replicate choice results using differential expression of the 8 signature genes to determine asthma endotype in kids. Obviously, doing this in kids is a big difference from the prior studies in adults. But we also wanted to see if we could do this using nasal epithelial samples obtained non-invasively rather than bronchial epithelial samples obtained using bronchoscopy. So we wanted to use airway samples instead of bronchial because it's just not that feasible to do a bronchoscopy, which is obviously really invasive and requires sedation on children in a big study. It wouldn't be feasible to do this in routine clinical practice for children with asthma either. Instead, we wanted to see if we could use nasal epithelial samples. So everybody here knows the nose is part of your airway and it's continuous with your lower airway, but it can be sampled far less invasively using a curette to scrape the inferior nasal turbinate, which you can easily access through the nostril. So to test if nasal epithelial gene expression could be used to determine asthma endotype in children, we used 3 independent studies of youth with asthma. The first is STAR, which is the Stress and Treatment Response in African American and Puerto Rican Youth with Asthma study. The second is EVA-PR, or the Epigenetic Variation in Childhood Asthma in Puerto Rican study. And the third is VDKA, or the Vitamin D Kids Asthma study. And in each study, we scrape the inside of each participant's inferior nasal turbinate to collect epithelial cells that we could then use for RNA sequencing. And you can see that there are a total of 459 children across the 3 studies that we included in our present study. Now, I want to highlight some important differences between these study populations. They were recruited from different places, Pittsburgh and Puerto Rico, and they are racially diverse. STAR includes youth who are Puerto Rican or African American. EVA-PR includes only Puerto Rican youth, and VDKA includes a variety of racial identities. This is important because asthma is not an equal opportunity offender. Asthma prevalence varies significantly by race. Individuals who identify as Black, Puerto Rican, or Alaskan Native Pacific Islander have much higher asthma prevalences than individuals who identify as white. Race is a social construct, so we suspect these differences in asthma prevalence largely stem from historical and structural racism. We believe that it's extremely important to include populations who bear the heaviest asthma burden in our studies of asthma, so that we can better address health inequities and ensure that scientific advances will benefit populations worldwide. You can also see that everybody in STAR was steroid naive. No inhaled, nasal, or systemic steroid use for at least 4 weeks before we collected their nasal cells. Conversely, everybody in VDKA received inhaled steroids prior to nasal sampling, while only about a quarter of those in EVA-PR received inhaled steroids. There was minimal use of nasal steroids in both EVA-PR and VDKA. And then none of the participants in any study received systemic steroids or had an upper respiratory tract infection prior to nasal specimen collection because we were concerned that exogenous steroids or viral infection could influence nasal epithelial gene expression and impact our endotyping results. So those were exclusion criteria. For more details about the children in our study from each of these 3 studies, STAR, EVA-PR, and VDKA, you can see Table 1 in our manuscript. So what do we find in these nasal epithelial samples from kids with asthma? As a reminder, we analyzed levels of expression of the 8 signature genes, periostin, SERPIN-B2, CLCA1, CSF3, CXCL1, 2, and 3, and IL-8. And then we did k-means clustering analysis to separate them into clusters based on their gene expression levels. So if you're following along in the paper, this is figure 2. We'll start with STAR, which is one of our 3 study populations. This is a heat map of expression of the 8 signature genes. And don't worry, for those of you not familiar with heat maps, I'll break it down for you. On the color key here, blue represents low expression and red represents high expression of a gene. And on the heat maps, each participant is a thin vertical line. And you can see 3 distinct groups of participants. The first group has red on the top, which signifies high expression of the 3 T2 genes, and blue on the bottom, which signifies low expression of the 5 T17 genes. So these are the T2 high individuals. The middle group here has blue on the top, which signifies low expression of the T2 genes, and red on the bottom, which signifies high expression of the T17 genes. And these are the T17 high individuals. And then the last group on the right has blue on the top and the bottom, which signifies low expression of both the T2 genes and the T17 genes. So these are the T2 low, T17 low individuals. So in STAR, we found that based on nasal epithelial expression of these 8 genes, children with asthma could be categorized into one of the 3 asthma endotype profiles. Then we did k-means clustering on the other 2 studies of children with asthma, and you can see we found really similar results. Again, there were 3 distinct profiles in each study: those with high expression of the T2 genes, those with high expression of the T17 genes, and those with low expression of both. So our findings suggest that nasal epithelial gene expression can determine asthma endotype profile in children. To validate our clustering method, we also employed alternative clustering methods, so we weren't just relying on k-means clustering. We also employed hierarchical, k-medioid, Gaussian mixture, and spectral. And after we used the 5 different clustering approaches, we found that 82% of participants in STAR had the same endotype across all 5 clustering approaches. 75% of EVA PR had the same endotype across all clustering approaches, and then 79% of BDKA had the same endotype across the 5 clustering approaches. So we deemed individual who had the same endotype assignment across all 5 clustering approaches to have a consensus cluster. So in other words, those are the individuals with a consistent, unambiguous endotype assignment regardless of which clustering approach we employed. And these are the heat maps of the individuals with a consensus endotype. And you can see the 3 endotype proportions in each study are very similar to what we found when we included the full sample with just k-means clustering. So this table is a little bit hard to look at, so I apologize, but I wanted to show you how the conventional T2 high biomarkers compared across the 3 endotypes in each of the 3 studies. For reference, eTable 4 in the online supplement to our article has a much more detailed version of this table comparing characteristics by endotype across the 3 studies. But you can see that atopy, which we defined as having at least 1 positive allergen-specific IgE, was significantly more common in the T2 high asthma group than in both of the T2 low endotypes in STAR and EVA PR, but it was still very common among both T2 low endotypes. At least half the participants in every endotype, even the T2 low endotypes, had atopy. And in BDKA, there was no difference in the prevalence of atopy among the 3 endotypes. Similarly, total IgE was higher in the T2 high endotype than in both T2 low endotypes, but there were plenty of T2 low individuals with what we would consider elevated total IgE in clinical practice. And same for eosinophils, highest among the T2 high participants, but it's still not really low among the T2 low individuals. So next, we wanted to see how well these T2 biomarkers that we commonly use in clinical practice identify the T2 high transcriptomic endotype profile. We did this analysis in STAR because this was the only study in which we had FeNO in addition to blood eosinophils and total IgE. So this is figure 3 in our paper. We used the Uden index to define the optimal cutoffs for individual biomarkers. The cutoff for eosinophils was 210.4. For total IgE was 417.5, and for FeNO was 32.5. When we applied these cutoff values, we found that 84% for total IgE and STAR were correctly identified, 69% for eosinophils were correctly identified, and then 75% of participants for FeNO were correctly identified. So for reference, for those of you who don't commonly prescribe asthma biologics, to be eligible for the T2 biologics that block eosinophils, a person needs to have an eosinophil count of at least 150. And you can see that the optimal threshold for eosinophils to identify the T2 high transcriptomic profile was significantly higher than 150. And on my prior slide, I showed that the median eosinophil count among the T2 low endotypes in all the studies was actually higher than 150. Similarly, to be eligible for omalizumab, the asthma biologic that blocks IgE, you have to have a total IgE of at least 30. but the optimal cutoff we found to identify the T2 high endotype was a whopping 417. And on the previous slide, you saw that the median total IgE in T2 low endotypes is much higher than 30. So I do wonder if this is why in my severe asthma clinic, some children don't seem to respond well to the biologics that they're eligible for. Like they have phenotyping labs that make them eligible for a given biologic, but they don't have improvement in asthma control after we start it. I wonder if I'm incorrectly categorizing those kids as T2 high. Like maybe they actually have a different inflammatory pathway driving their asthma, and that's why they don't respond to T2 high biologics. Once I'm done presenting the article, I'd love to hear the panelists' thought on this hypothesis or theory. We were also really excited to see that these 3 endotype profiles occurred in similar proportions across the 3 studies, despite the differences in study populations that I highlighted earlier. Of note, T2 high asthma only occurred in 23 to 29% of children. And as I mentioned before, it's generally thought that T2 high asthma is the most common type of childhood asthma, but our findings and findings from some other recent studies are starting to challenge that assumption. Our finding that T2 low asthma is the predominant type of childhood asthma makes it even more urgent for us to better characterize and develop treatments for T2 low asthma. So next we did a differential expression meta-analysis. We did this for T2 high asthma, comparing that group to the T2 low, T17 low group, and then also for T17 high asthma, again comparing to the T2 low, T17 low group. And doing this allows us to identify other differentially expressed genes for the T2 high and T17 high endotypes. So these are the differential expression meta-analysis results for the T2 high profile, and this is figure 4 in our paper. We found 3,516 differentially expressed genes for the T2 high profile. And as expected, by our definition, periostin, SERPIN-B2, and CLCA1 were upregulated in the T2 high profile, but you can see that there's a lot of other genes that were also upregulated in T2 high compared to the T2 low, T17 low individuals. And then these are the differential expression meta-analysis results for the T17 high profile. We found 2,494 differentially expressed genes for the T17 high profile. And as expected, by our definition, the 5 T17 signature genes, CXCL1, 2, and 3, CSF3, and IL-8 were upregulated in the T17 high profile compared to the T2 low, T17 low profile. But again, there are a lot of other genes that we identified that were upregulated in T17 high. You can see that CL6, which attracts neutrophils, is upregulated in the T17 high profile, as is IL-23A, which is also strongly associated with T17 inflammation. And then these are the results for the canonical pathway enrichment analysis we did using differentially expressed genes that we found for the T2 high profile, which are shown on the left here, and then the T17 high profile shown on the right here. This is figure 5A and B in our paper. And you can see that the highest enriched pathways for the T2 high profile were related to cellular signaling, and then the highest enriched pathways for the T17 high profile were related to neutrophils and, unsurprisingly, IL-17. And then finally, these are our results for the gene ontology analysis on select gene modules that we identified for the T2 and T17 profiles. You can find a lot more details about these in the online supplement. But glycoprotein-related pathways and IL-13 signaling were upregulated in the T2 high profile, while pathways related to viruses, cytokines, and interferon gamma were upregulated in the T17 high profile. These differential expression, canonical pathway enrichment, and weighted gene co-expression network analyses identify genes and pathways that warrant further investigation for the T2 high and T17 high endotypes. So this study has several limitations. First, there's no gold standard to define asthma endotypes, so it's really hard to definitively say how well our transcriptomic endotyping method actually performs. However, we tried to validate our findings using existing T2 biomarkers, as well as examining upregulated genes and pathways associated with T2 high and T17 high profiles. Our analyses showed that T2 high transcriptomic endotype profile is associated with existing T2 biomarkers, as well as pathways that have been previously identified to be associated with T2 inflammation, like IL-13. And then similarly, the T17 high transcriptomic endotype profile was associated with pathways related to T17 inflammation. Second, assignment to a transcriptomic profile was based on a single measurement, and endotypes might change over time. We don't really know. So further study will be needed to determine the stability of endotypes over time. Third, individuals may be able to have more than one asthma endotype simultaneously, but the clustering approach that we used in this study does not allow for individuals to belong to more than one endotype. So again, further studies with different diagnostic approaches will be needed to try to determine if these three endotypes are actually mutually exclusive. And then fourth, nasal epithelial transcriptomics, although they've been well correlated in prior studies, may not fully reflect bronchial epithelial transcriptomics. And then finally, this study included mostly Puerto Rican and Black youths, so our results may not be generalizable to youths of other races or ethnicities. So in sum, we found that indeed, nasal epithelial gene expression can be used to determine asthma endotype profile in kids. We also found that the T2 high nasal transcriptomic correlates with having high T2 biomarkers, but lots of T2 low individuals also had high T2 biomarkers. T2 high asthma was not the predominant asthma endotype in our study. Actually, less than a third of participants had T2 high asthma using this method. And then we identified multiple differentially expressed genes for the T2 high and the T17 high endotype profiles, which warrant further investigation and may represent novel therapeutic targets, which is, of course, of particular importance for T17 high asthma, given the lack of existing treatments for that group. So I want to acknowledge my co-authors, particularly my co-first author, Len, as well as Dr. Celedón and Dr. Chen, who were the co-senior authors on the study, and then the research coordinators who helped make this study possible. And with that, I'll turn it over to our panelists.
Great, Dr. Gaietto, that was absolutely fantastic. And this is such wonderful work. And I'm very curious to learn what our panelists have to share with their impressions and insights as well. So with that said, Dr. Barcharier, would you like to kick things off?
Sure, thanks. Kristina, that was wonderful. And you and your colleagues should be congratulated for really a very important piece of work that really sheds some light on the pathogenesis of childhood asthma. Much of this space has been consumed by our adult colleagues, and we all know that children are not little adults. Adults are just big children. But you know, one of my questions really has to do with that element, which is that you, because you really didn't have any other choice, you used the genes that the adult folks have used to define type 2 high. And, you know, maybe that's part of the explanation here, is that the genes responsible for driving type 2 inflammation in children may be a very different collection of genes than what have been described in adults. You know, in our study with mepolizumab that I think you alluded to, we found a very different response in children than had been seen in adults. And it made us question the relevance of at least that pathway in children. So, you know, on your volcano plot, there are some genes that are really very highly expressed, yet weren't in your initial categorization. And I'm wondering if you think, had you done it again, knowing the answer before you started, would you have defined type 2 high with that same 3 gene signature, or would you have used others that might be potentially more relevant or specific to children?
I think that's a fantastic question that you raise, and I'd love to hear Dr. Celedón's thoughts as well. But I will say, I think as a preliminary first step, just like to show that there's some validity to what we're doing, I think we have to look at what's been done already in the adult population. And then as you said, I think there are potentially other ways, better ways that we can define the T2 transcriptomic profile in youth based on what we found using, you know, this clustering approach and then looking at other like differentially expressed genes, other upregulated pathways. So that's definitely some future studies that we're working on right now. But you raise a really excellent point. I think it all comes back to currently there's no gold standard to define these endotype profiles. And so it's really difficult to say how like reliable this is without having a gold standard to test it against.
Agreed, and you started in the right place, and the question is the next pass through, knowing what we know, might it be done differently? The other point I wanted to make is when you were showing those initial heat maps, what struck me is that it's going to one of your concluding points. I don't think these phenotypes are mutually exclusive. I think most people have a complex endotype. They're, especially those who have the type 2 high signature, they still got a lot of TH17 activity in the background. It just gets overwhelmed by the type 2. In contrast, those who have a lot of TH17 activity tend not to have a lot of type 2. So maybe that's the reason the clinical responses are as little less predictable than you think, is because almost everybody has some component of a mixed endotype. And, it's trying to figure out which is the dominant one, which is driving that individual patient's disease. And, maybe that's why we get it wrong sometimes clinically, that we think it's the eosinophils because those are the highest counts. So we treat them with an anti-eosinophil drug and they don't get better. It's because there was a second, either type 2 high or non-type 2 pathway that's really the dominant, and that's the reason we lack clinical response. But congrats on a really terrific piece of work that I think gives us a lot to think about.
Thank you.
Thank you, Dr. Barcharier. Dr. Rastogi.
Yeah, again, just to repeat, I mean, it was fascinating reading this paper, and as a clinician, I enjoyed it so much because you took what was almost, signature agnostic. You had an excellent set of populations, took 3 populations simultaneously, and investigated this in a population that needs to be understood better. And then you took it to the clinical, parameters we think about every single day, and then you brought it back again to say, okay, now that I've defined this, which other genes can I identify? And how beautifully you kept, verifying and validating your findings. So congratulations again.
Thank you.
Along those lines, you touched upon the utility of the cutoffs and, what we as panelists think and how much higher were your actual cutoffs. And then, it makes me wonder, and a lot of us, on this call work in the severe asthma area, where maybe what we're working with is not good enough and much higher values should be thought about, or maybe our population who we have some reason to think about them, maybe not being so T2 high, maybe the ones that we don't immediately jump towards doing biologics. But even before that, can we be steroid sparing in these populations? You've touched upon obesity and this is my bias that I'm coming in with over here. Is that a space where maybe our IgE and our FeNO and our other measures are not that high by the standards that are routinely used, but maybe not in our patient population? And, it makes me think about the cutoffs we use, say the trypsinogen for CF, where you have a bell-shaped curve, you have a Z score for your population, and then you're able to say, okay, fine, if you're in that other end of the spectrum, the highest, 95th percentile, or whatever cutoff you would like to use to extend. So just to pontificate a bit on the clinical thoughts that I had around this. And then, coming back to obviously having the feasibility of accessing what is closer to the airway than reaching the airway itself in the patient populations that we take care of. So I was thinking as I was reading this paper, about other potential nasal involvements such as non-allergic rhinitis or vasomotor rhinitis. And, you obviously have a wealth of data in these studies, and maybe the implication in the non-allergic realm wasn't being thought of when these cohorts were recruited, but I'm curious how would you now moving forward think of these signatures in the realm of what may be nasal pathology along with a physician diagnosis of asthma?
Yeah, I think that's a very great question that you raise. We did collect information on like history of allergic rhinitis and some of those other comorbidities that you mentioned. We also looked at like seasonality in case that was playing a role into this, and we didn't find anything significant, but you're totally right. Like we don't exactly know, and we also don't know what we didn't measure, if that makes sense.
And then pulling it to, how we recruit our cohorts, that this wasn't associated with lung function. So was lung function highly preserved in these cohorts? And then would it make sense to almost extend it in the severe asthma group again, where frequently and more often than not, we cannot get their lung function in normal range. So even if we were to study the subset who are the worse off by however limited our lung function quantification is, but are, again, in that lower, 5th percentile or 15th percentile or whatever, to consider looking at some of these genes specifically. And then the last question I had, obviously compartments, we're looking at the nasal compartment, how much of this can we measure in the blood, because that may be potentially more easily accessible. Processing is better established for that rather than the nasal sample. So if one were to think of immediate translation to the extent one can in a clinical space, then, if you have access to those profiles in these cohorts, even looking at the concordance versus discordance in maybe a compartment which is more easily accessible.
Yeah, I mean, I'm biased, but I think that's an excellent question that you raise. Dr. Celedón is looking at gene expression in the blood, and I'm actually really interested in potentially like the protein products of these in the blood. And I submitted a KO8 to look at that to see if we can find blood protein markers of the T2 low endotype. So yeah, I like your thought.
Excellent. Glad to hear that you're all thinking along the same lines. So now this is all I had, and congratulations again. Beautiful, beautiful manuscript.
Thank you.
Thank you so much, Dr. Rastogi. And I can see that Dr. Tantisira has been shaking his head throughout your comments as well. So please take it away, Dr. Tantisira.
I love the fact that you've submitted a case, so congratulations on that, Kristina, and particularly in a very, very relevant area. So best of luck to you on along those lines. I just wanted to say again, tremendous job. I wanted to actually reiterate a couple of things that I really, really liked about this study. I love the fact that as a first study, you actually didn't go straight to the blood, but you actually are using nasal epithelium because I do think that it's nice to have a airway epithelial cell of some sort as part of the paradigm process. I also love the fact that you have replication across not just one but multiple cohorts, and so I think for everybody who has not directly involved in omics type of studies before, one of the hardest things to do, even though you mentioned generalizability as a limitation, you're actually demonstrating generalizability, and you really should be applauded for that in terms of your overall efforts. And then finally, I think, the fact that you have something that's compelling in terms of potentially paradigm shifting, in terms of saying, oh, well, it's actually potentially just a small portion of individuals that are truly T2 high. Granted what Len said and others have mentioned about maybe mixed phenotypes and things along those lines. Before I have a couple of questions, but before I get to those, I also wanted to make one additional comment. I know that you had specifically mentioned, oh, well, I have a set of patients where I believe they should respond to biologics and they meet clinical phenotyping. But yet they don't seem to do as well as I would hope to, I mean, either non-responders or partial responders. And I think any of us who are involved in severe asthma clinics see a lot of this non-response or partial response compared to clinical biomarkers. I will also throw out to you that the converse is true, although we actually don't do this because we have biomarker-driven therapies. So in theory, we're not supposed to be giving medications to people who don't meet biomarker criteria. But there have been, including some data that I've seen on post hoc analyses of benralizumab, for instance, not published, but there's approximately 45 to 50% of individuals in the low eosinophil group that actually have a meaningful clinical response when measured as can you reduce exacerbations. And so I think we're actually dealing with two different things here. We have a group of patients who theoretically should respond based on biomarker criteria, and we have a group of patients who theoretically shouldn't respond based on biomarker criteria. And I think the bottom line is that we need better biomarkers, right? And we also need to better understand the biology because, in my example for benralizumab, it's not just the eosinophil that is doing this, right? And so I think that your study is starting to make a foray into that. And so I'm excited about that, but I think that we could go further in terms of not just using your exemplar genes. And I think piling a little bit onto what Len started off with is you have a whole new different set of genes that you could say, oh, well, these could be T2 high genes. What is their potential role in terms of can we come up with an alternative signature that might better define what could be a dupilumab or a benralizumab or a mepolizumab type of T2 high response biomarker. So I think again, all of this is just scratching the surface. We've got a long way to go, but I firmly believe in this approach to try and identify better molecular biomarkers that are not the typical clinical biomarkers that we're using for every day. Do you respond or do you not respond to one of these biologic therapies? I'll just close with my two quick questions. One is, I kind of liked your approach where you kind of did a kind of a pseudo supervised but not supervised clustering. So, you kind of defined a set of genes. So that's the supervised portion. And then you did the unsupervised portion where you clustered individuals together. I think that traditionally folks have kind of gone one extreme or the other. One is they've either started with clinical endotypes, right, to say, here's all the clinical characteristics we have. What are the differentially expressed genes therein? Or they've gone the whole other way and just said, oh, well, we've got 20,000 genes in an RNA sequencing. Let's see how those cluster together just by themselves, independent of doing stratifying by pre-existing knowledge of what might be T2 high or T2 low. Have you done either of those alternate approaches to your clustering? And if so, what did you find along those lines?
That's a great question. So I personally haven't done either of those approaches. I'll let Dr. Celedón comment on if he or others in his lab have been working on those specific questions. I will say that the approach of like using the defined signature genes is where we started on this pathway. And then maybe I'm like saying too much, but based on reviewers' comments during the multiple iterations of revisions for this paper, we ended up doing that sort of more like broad clustering approach based on differentially expressed genes. And in the end, I think we all really liked what we found. Like we liked that approach and liked the result. But that was the result of multiple iterations of revision.
Excellent. A lot of wonderful questions and generating wonderful discussion, which we really appreciate. Last but not least, Dr. Celedón.
Yeah, no, I wanted to say the comments from all panelists were very thoughtful and right on, line with what we're thinking. So let me start with Dr. Barcharier's comments. I think they are very relevant. There is no doubt in my mind that some of these endotypes will overlap, right? And time will tell. The other thing that Kristina mentioned is that I also predict that they will change over time. There is minimal data on this. The one epidemiologic study that I've seen from Germany showed that over a period of 2 years, about 20% switch endotypes, right? So that's a question we don't know. The other question is how will treatment, impact these endotypes, right? All these kids were off treatment as best as we could tell, but what happens if you have a kid with T2 high and you treat them, and how does the endotype look like? So I think those are all great questions. we have used, different sets of genes to define a T2 high. We've also used, I'm sure many of the people in the call are familiar with a study by John Fahey's group in sputum. So unlike us, they didn't use T17 high. They couldn't, in sputum. They used T1 high. So we've taken a preliminary pass at that. All I can tell you so far is that it doesn't change the proportion of people with T2 high. So it's still low. That, as you would imagine, some of these, and that's a limitation they discuss, some of the T1 high, in fact, have T17 high, right? So I don't think that would surprise anybody, in the field. But we're also looking at that. So, I think it's a proof of concept and it's a first pass. There is still some work to do. But that said, I think that one of the first initial applications of this, and I hope that it will be used that way, hopefully, is to try to have better targeted trials. So I think, as imperfect as this may be, if we can enroll kids and finally be able to differentiate T17 high, going back to Dr. Rastogi's comment on obesity, right? I think that would be important. The other application of this that is coming down the pipe, and there are already several papers that are either accepted or under review, is of course look at risk factors for these endotypes. Prior to the analysis that are ongoing, my prediction, I think most people would agree, would be that some of these risk factors may be pan-endotypic, if you will. So they may affect all endotypes. But what we're seeing right now, and I can mention the first study because it's about to be accepted, I hope, is on obesity. And as Dr. Rastogi probably would predict, no surprise to us, it was associated with predominantly T2 low asthma endotypes, right? So T17 and low. This was true for obesity or overweight. It was true for biomarkers of adiposity. So that's just one example. So I think you'll see more of this, coming up. And in terms of other things that were mentioned, the severity question is a great one. So my counterpoint to studying kids with severe asthma is that in children, only 5% of asthma is severe, right? As everybody in the call know. So I think we're actually neglecting by virtue of the concentration on severe asthma, the vast majority of children who have mild to moderate asthma, right? And are usually not included in these, studies of endotypes and so forth. So I think they are very important. And then, I agree entirely with trying to correlate the findings with blood. And that's something that Kristina, intends to do. We have other people working on this. I'm also excited about another paper coming up, which is correlating DNA methylation with the endotypes. Because DNA, as all of you know, is more resistant also, more resilient than RNA. So, we'll see how that works. And then finally, I agree with Dr. Tantisira's comment about therapies. I think that would have to be a multi-center study, Kelan, for sure, because it's so hard to get an accrue enough number of kids on these biologics. But of course, that would be another great sort of question to interrogate. So very thankful for all the comments. Thank you.
Right now, there are no questions in the chat, but I just wanted to ask, congratulate Kristina on such a great presentation and the insightful comments of our panelists. But Kristina, you mentioned it a bit in the paper, but can you just summarize quickly that they were in the supplement, the pulmonary function testing across your different endotypes and the response to ICS in the different endotype classifications that you found?
Yeah, that's a great question. So we didn't see significant differences in lung
That's what I
function
thought.
across the endotypes. Yeah, or we only had ICS response data for the STARS study because those children like ended up getting inhaled corticosteroids after we did the nasal sampling for a period of 6 weeks, and then we were able to look at their ICS response over 6 weeks, and we didn't see differences by endotype.
Well, let me expand on that a little bit. So, as Kristina mentioned, so only STAR had data on ICS response. So very small groups. So this is why it wasn't a major point in the paper. But what we did find is for that T2 high group, there were some predictors. So a high FeNO. Now, when I mean a high FeNO, it's a really high FeNO. So it wasn't 20, it wasn't 30, 50 was sort of the cutoff where you saw responses. Not unsurprisingly to everybody, the kids who experienced the highest drop in FeNO between baseline and 6 weeks of treatment also, were in the responder category. So those were the major findings. For the low endotypes, the predictor of response was low FEV1 which is kind of interesting, right? A pulmonary function test. But again, we had no replication of that. And I can tell you that JAMA reviewers and editors appropriately were brutal on that we had no replication, and therefore, that's why it's in the supplement. But since you asked, I share with you the results. So thanks.
Thank you so much. We are getting close to the hour. I don't know if Annie has any final comments, or do you want to see if there's any other things you want to conclude with?
Sure. I guess before my final comments, are there any other comments or questions from our panelists or any of the participants on the call? Looks like not at this time. Again, Dr. Gaietto, thank you so much. This was absolutely a fantastic presentation and such a lively discussion. So this concludes our journal club on transcriptomic. Whoever knew it was that hard to pronounce, profiles and asthma endotypes in pediatrics. I want to thank Dr. Gaietto again and all of our panelists, my co-moderator, Dr. Santiago, and Sam Carpin for her tech support. A recording will be posted on the ATS Journal Club site. It's typically up within about a week of our sessions, and we welcome any of your proposals for future interesting articles and or to present future journal clubs. If this is something of interest to you, please consider contacting myself or Dr. Santiago or Dr. Mitzi Go with your suggestions or any questions that you may have. But thank you all for your participation.
Thank you very much.
Thank you very much.
Thank you.
Great.
Bye.